Hello everyone, welcome to Dentistionary. Today we are back with our second lecture in human physiology in the unit respiration, and our topic for today is transport of gases. Mark this topic as one of the most important questions asked in terms of your prof or in terminals. In transport of gases we will learn about how oxygen is transported in our body as well as the carbon dioxide. So let's begin. Oxygen transport. We all know oxygen is essential for ATP generation through oxidative phosphorylation, and therefore it must be reliably delivered to all the body parts. Absence of oxygen may lead to hypoxia. Proper description of hypoxia will be done in our further slides. Blood becomes typically saturated with oxygen after passing through lungs, which have a vast surface area and thin epithelial layers that allows for the rapid diffusion of gases between the blood and the environment. Oxygenated blood returns to the heart and is distributed throughout the body by way of systemic vasculature. Oxygen is transported in two major ways. First, in dissolved form, and second, as oxyhemoglobin. What is oxyhemoglobin? When oxygen binds with the heme part of hemoglobin, it forms oxyhemoglobin. So, about 3% of oxygen is carried in dissolved state through plasma, and remaining 97% of oxygen is transported by RBC in the blood. In a normal person, the hemoglobin level is about 15 gram per 100 milliliter. The capacity of 1 gram of hemoglobin to combine with oxygen is 1.34 milliliter. Therefore, arterial blood carries about 20 milliliter oxygen per 100 milliliter of blood. When it comes to the binding side, each hemoglobin part carries 4 molecules of oxygen. So, approximately 15 milliliter of oxygen is transported by hemoglobin during exercise, and under normal condition it is 4.4 milliliter of oxygen. These all are being calculated in terms of 100 milliliter of heme. Next important topic is the oxygen dissociation curve. What is oxygen dissociation curve? It is simply a graphical representation between the partial pressure of oxygen and the percentage saturation of hemoglobin with oxygen. It is represented in the form of S or the sigmoid curve. Wait, let me show you the diagram. Yes, this is the oxygen dissociation curve. You can see it is S-shaped. The blue line represents the left shift of oxygen dissociation curve, and the green line represents the right shift of oxygen dissociation curve. Now the question arises, why these are being shifted? Yes, when oxygen dissociation curve is shifted to left, it occurs due to the following causes. Number 1, high partial pressure of oxygen, low partial pressure of carbon dioxide, less H plus concentration, that means higher pH, and low temperature. If you ask for an example, fetal hemoglobin is one of such type whose graph will be shifted to the left. Myoglobin is another example. And when it comes to the right shift, it occurs due to the low partial pressure of oxygen, high partial pressure of carbon dioxide, high H plus concentration, low pH, and high temperature. Hope so this much is clear to you all. This graph is very important. Please add it to your answer. Next, we have the transport of carbon dioxide. When we saw in terms of oxygen, it has been transported in two forms, in dissolved state and in oxyhemoglobin form. But in terms of carbon dioxide, it has been transported in three major forms. In dissolved form through plasma, about 7% of oxygen get transported. It gets dissolved in the blood plasma and is carried in solution to the lungs. Next, in bicarbonate form, about 70% of carbon dioxide is converted into bicarbonate ions and transported in plasma. Oxygen diffuses in RBCs where it binds with water, forming carbonic acid H2CO3. Carbonic acid quickly dissociates to H plus ions and bicarbonate ions as it is unstable. You can see the reaction. CO2 plus H2 gives you H2CO3, which dissociates to H plus ion and HCO3 negative ion. Next, it is also been transported by RBC as carbaminohemoglobin. Hemoglobin with carbon dioxide forms carbaminohemoglobin. Please don't get confused with carboxyhemoglobin and carbaminohemoglobin. When carbon monoxide binds with hemoglobin, it forms carboxyhemoglobin, whereas when carbon dioxide binds with hemoglobin, it forms carbaminohemoglobin. In terms of carbaminohemoglobin, about 20 to 25% of carbon dioxide is transported. Carbon dioxide that enters in the RBC forms a reversible compound, that is HbCO3. This CO2, I'm sorry, it's HbCO2. This CO2 binds with amino group of globin part, which is a part of hemoglobin. The reaction is similar to the oxygen, but carbon dioxide binds not with the hem part but with the protein part of hemoglobin. Right? Is it clear? This is the carbon dioxide dissociation curve, same as oxygen dissociation curve. Next we have the chloride shift. Mark this as very, very important. Chloride shift is also known as hamburger phenomena. So what is chloride shift? It is basically a process which occurs in a cardiovascular system and refers to the exchange of bicarbonate ions and the chloride ions. Across whom? Across the membrane of the red blood cells. First let me show you the diagram. Yes. So this is the proper diagram representing the chloride shift or the hamburger phenomena. Next what we have, why this hamburger phenomenon occurs. The chloride shift or the hamburger effect describes the movement of chloride into the RBCs, which occurs when the buffer effect of deoxygenated hemoglobin increases the intracellular bicarbonate concentration, and the bicarbonate is exported from the RBC in exchange for chloride. बाइकार्बोनेट आयंस एंड क्लोराइड आयंस के बीच में जो भी एक्सचेंज होता है दैट इज कॉल्ड द क्लोराइड शिफ्ट और द हैम्बर्गर फेनोमेना. The bicarbonate ions diffuses out into the plasma and the chloride ions enter into the RBCs at the level of tissues. This is known as the chloride shift as already mentioned. During external respiration, chloride ions move out as the bicarbonate ions move in. This is called the reverse chloride shift. एक्सटर्नल रेस्पिरेशन के वक्त यही प्रोसेस उल्टा हो जाता है एंड दिस इज नोन एज रिवर्स क्लोराइड शिफ्ट. क्लियर? नेक्स्ट देयर अनदर important topic called the Haldane effect. I have forgot to mention in this slide. What is Haldane effect? It is basically related to the transport of carbon dioxide in the blood. It is based on the simple fact that oxyhemoglobin behaves as strong acid and releases an excess of H plus ions which bind with the bicarbonate ions to form carbonic acid, right? H plus ions means the H plus binding with HCO3 negative, that is the bicarbonate ion, to form H2CO3, that is the carbonic acid, which dissociates further to form H2O and CO2. Why? Because it is highly unstable. Now, due to the increased acidity, this CO2 loses the power to combine with hemoglobin and form carbaminohemoglobin. So, the effect of oxyhemoglobin formation or the dissociation on carbon dioxide transport is called the Haldane effect. Simple, clear? ऑक्सीहेमोग्लोबिन फॉर्मेशन कार्बन डाइऑक्साइड ट्रांसपोर्ट के ऊपर जो भी इफेक्ट देता है मींस द इफेक्ट ऑफ ऑक्सीहेमोग्लोबिन फॉर्मेशन ऑन द कार्बन डाइऑक्साइड ट्रांसपोर्ट is basically called the Haldane effect. Hope so this much is clear for you guys. There's always come a question, write the difference between Bohr's effect and Haldane effect. Haldane effect तो clear हो गया. Now what is Bohr's effect? Bohr's effect is just the reverse of it. A rise in partial pressure of carbon dioxide or the fall in pH decreases the oxygen affinity of hemoglobin. Clear? पार्शियल प्रेशर में कार्बन डाइऑक्साइड जब बढ़ जाएगी तो हेमोग्लोबिन के साथ ऑक्सीजन की बाइंडिंग कम होगी, रेजिंग द वैल्यू, राइट? दिस इज कॉल्ड द बोर्स इफेक्ट और इसी के उल्टे को क्या बोलते हैं? हेल्डन इफेक्ट. होप सो दिस मच इज क्लियर. आज के लिए इतना ही. थैंक यू सो मच गाइज. In our next slide, we'll discuss the further topics related to respiration. Till then, keep supporting our channel and keep showing your love towards Dentistionary. Thank you.